are deeply rooted in pulmonary diseases including asthma, lung fibrosis, and cancer, with a special focus on the role of nuclear receptors and natural products and genetic associations in disease mechanisms. His work has been widely recognized and supported through competitive research grants from prestigious national funding agencies such as CSIR, DST, SCRB, DBT, and UGC, as well as private sector collaborators like Virtue Biotech. Till date, Dr. Reddy has authorized 4 books and contributed to 10 book chapters and published over 60 peer-reviewed scientific papers. He guided multiple PhD scholars with several ongoing doctoral projects under his supervision. Beyond research, Dr. Reddy is a dedicated academic leader and administrator. Sir served as the head of the Department of Zoology, chairman of the Board of Studies, and coordinator of the NSS cell at Yogi Berra University. His exceptional services to science and educational has been recognized with several prestigious honors like the Andhra Pradesh State Teacher Award 2022 by the Government of Andhra Pradesh, the Andhra Pradesh Scientist Award 2020 by APCOST, election as a Fellow of Young Academy of India 2021, and Sir is a editorial board member, SCCSCA nominee for IAEC, and the principal organizer of numerous workshops, seminars, and conferences, playing a key role in the academic outreach and capacity building. Dr. Reddy is not only a profile researcher but also a passionate teacher and as an inspiring role model for young scientists. With this brief introduction, I would like to welcome Dr. Venkatram Reddy sir to take over the session.
Thank you, madam. How are you, madam? Nice introduction. Good morning to all. వినిపిస్తుందా? కనిపిస్తుందా? మీరంతా pharmacy students, right? You are next to the doctors and the best to the doctors. You are the best providers for the doctors, right? So you are in a great profession, and very good morning to once again to all the participants. I thank the organizers for giving me this opportunity to share some of my experiences and thoughts regarding the IAAC, particularly Form B, which is very important for conducting animal research. It is very essential also if you want to really conduct any kind of research using animals or laboratory animals, which is very much essential. Okay, so that is why the organizers has given me this important topic. So as madam introduced, I am from Yogi Berra University, Kadapa. I have a very long association with this institute, RIPER, as one of the main nominee of IAAC for the last 7-8 years, and for 5 years I served as a CPCA nominee for 5 years, and then we have little bit collaboration also for doing some kind of research. My PhD students take the help of pharmacology department, in silico docking and other things. So it is very fortunate for me to be here. Once again, I thank the organizers for giving me this opportunity. So before we go to discuss the Form B, we will see what are the different essential things in the IAAC and the source of website. Many of the people think that Form B is a bureaucratic step. It is a document, but this is not just a document. So it deals with the ethical issues transparency, scientific validity of the experiments conducting on the animals. Okay? Scientific validity will be given by the evaluating of this form. While doing that, we also consider a lot of things. Whether the animal is going any kind of pain, distress, or any suffering. So these are the main aims of filling up these forms. Okay? So we have to minimize the suffering, we have to minimize the pain, and we have to minimize the distress of the animals while doing the experiments. So if you want to really do all these things, you have to critically follow the form B, and you have to understand each component of the form B, and then you have to start the experiment. Without understanding, without knowing what are the components present in the form B, you should not start any animal experiments. That is the important things you have to keep it in mind. Particularly students, when you do some kind of research at B pharmacy level or at M pharmacy level, you people are least bothered about the form B. Okay? This will be submitted at the last minute with a lot of mistakes that we have observed in many meetings. So while discussing, if you get any kind of questions or queries regarding how to fill this form, what to fill in this form, what kind of questions will be generated during the IASC meetings, all these things you can feel free to ask at any time of my presentation or after my presentation also. Okay? So before we going into the things, so this is the source for different forms, you can find it. So if the source is, this website is, you are very familiar now. Yesterday, Dr. Suresh was spoken about lot of things about what is CCSCA, what are the ethical issues, what is the IASC composition, what we do, all those things was nicely explained by the Dr. Suresh sir, and today also you might have learned a lot of welfare issues. So in science, there are two things when you use animals. One is scientifically justification you should have to do some experiments using animals. The second one is animal welfare issue. Without doing any issues related to animal issues, if you do science, that is not good. Okay, you have to balance animal welfare and the progress of the science balance. So that is the important things you have to do. So when you go to this website, you will find lot of forms related to various things. Okay, so we will see those forms first before we go to explaining of the form B in detail. The form A is application for registration of the institute. If any institute want to conduct animal experimentation, it has to be registered with CCSEA. So without registration, you should not conduct any animal experimentation. If you want to register, you have to fill this form along with all evidences and submit to the CCSEA. Then they will go through the documents and they will send one of the nominee to inspect the facility, and then if inspector is satisfied with the facility, then only they will approve the animal house facility with the registration. Then only you can do experiments along with the IAS, Institutional Animal Ethics Committee members also will be given. This we will discuss later. And Form C is record of animal breed acquired. So this is generally meant for the establishments. They have the breeding facility. Okay, breeding coordinates they import, they have to maintain these records. And Form D is very important. It has to be maintained by the principal investigator. Form D is nothing but the number of animals is approved in the IASC, and then number of animals utilized is a kind of utilization certificate. It has to be maintained by the investigator. While presenting the progress in the IASC meeting, it has to be submitted to the IASC for showing the progress, whether experiment is conducted or not, how many animals has been utilized, all those things has to be maintained. And Form E is record of animals. This is also maintained by the establishment, particularly when you have the breeding facility. Okay, so breeding facility persons can sell the animals. So how many animals they sold, sell, all these details will be maintained. So out of all these things, we will discuss this because this is very important for investigator perspective, because student can be an investigator, faculty member can be an investigator. So that is why it is very important for you guys to understand this. So this Form B has several objectives. So what are those objectives? The main objective is to ensure ethical use of animals in scientific research. Ethical means whenever you need animals, in that case only we have to use. Because you are doing some pharmacy work, project work, you should not use animals. If it is really essential, if it is mandatory, then only we have to use the animals. For that purpose, this Form B is one of the objectives. So once you fill the Form B, the committee will scrutinize whether it is essential or not. So it is also a document of complete information. So that contains what is your study, title, what kind of animals used, how many numbers, experimental designs, methodology, procedures, what dose, how many doses, all those things will be containing in this document. Okay? So it has every information about your research. And another important is legal and regulatory requirements. So this document is found under Rule 8 of Breeding and Experiments of Animals. PCA Act 1960 lo breeding and experiments of animals rules lo, Rule 8 is the Form B. So what this Rule 8 says that all experiments are approved by the IAAC before conducting any kind of experiments. So that rule has to be followed by every investigator. And also it promotes 3 principles. So you might be knowing all these things. Yesterday might have discussed. These 3 rules are very important when do the animal experimentation. Replacement, use alternatives to animals. Means you can do in vitro work, in silico work, other biochemical experiments if it is available. If it is compulsory only, you have to go for animal experimentation. And reduction, use the minimum number of animals. Instead of using hundreds of animals, you can reduce the number of animals. So that is one of the principles of refinement. So the refinement is very important things. Where we play important role in IAS. Refinement mainly to reduce the pain, suffering, and also distress of the animals. Okay. So we will also see what is the painful procedures you are doing, anesthesia are giving or not, how you are dissecting the animals, how you are euthanizing the animals. So those things are very important to minimize the pain, distress and suffering of the animals. Thus that comes under the refinement. And is assess scientific and technical justification. So these are some of the objectives. And it is a standardized protocol also. There will be a uniform structure will be provided in the Form B, so that committee will be easily go through that, and then they will evaluate, and then it can be a audited record. Any problem? వినిపిస్తుందా? ఫాలో అవుతున్నారా? డిస్టర్బెన్స్ ఉంది బ్యాక్ సైడ్. Okay, so these are the important objectives. Come to the Form B. Form B, as I mentioned, it comes under the Rule 8A of Breeding and Experiment of Animals under PCA Act 1960. So this rule says, without filling of this form and submission of this protocol to IAAC, you should not conduct any kind of animal experiments. So it has 2 sections. The first section, one is general information. It is a very straightforward. And second section is important. So that contains many of the questions related to animals, how you are handling animals, how you are dissecting animals, methodology, discard, all those things will be given in the part 2 or section 2 of the things. So this rule, as I mentioned, this rule comes under the breeding and the experiment of animals under control and supervision rules or PCA Act, Prevention of Cruelty to Animals. So animals like human beings, they suffer. So that is why this act came, Prevention of That was 1960. And later this rule is amended in different years. Okay. So what this rule says, in application form B, if you see on top of that, you see this rule 8A, form B, rule 8A. So that you can see. So that is the meaning. What is that meaning? What this rule says that requirement of prior permission from the institutional animal ethics committee for conducting of any kind of experiments using animals. So this rule we have to follow. Otherwise, you are deviating the guidelines of CCSCA, and it can be punishable also. Any experiments of animals, if you do without the knowledge, without the information provided to the CCA, is offensible, punishable. Okay? And what are the main provisions that is present in this rule? One, no experiment shall be performed on the animals without the permission of the IAAC. Permission must be obtained by submitting this form, and you have to take the approval. And IAAC will scrutinize this protocol and recommend you to conduct this project. Or if there is any lacunae, any issues, they will ask you to revise the protocol. So these are the important provisions that is present in the things. So now we will go into the form B sections. So form B is printed and distributed among you, right? Is that the form B? What are the form B? Ichara saru? Ichamanare saru? Okay. So the form B, if you have, you can go through it while I am explaining what are the each component, or you can directly watch from this here. Okay. So this form B, section 1 contains 7 questions. So these 7 questions are straightforward questions, and it is related with you, that means the principal investigator, institute registration number, and where the animals will be procured, that address it is contained. Okay. So you have to sign here name and designation of the investigator. Mainly the principal investigator should sign, not the students, not the postdoctoral fellow or any other person. If the postdoctoral fellow has any specific funding from DST, DBT, CSIR, that means independent funding, they can submit as an independent researcher. He will be postdoc, can be considered as a principal investigator. So we will see the details in the form B, section 1. Name and address of the establishment, registration number. It has 7 questions. Directly we will go to how to fill these 7 questions. Name and address of the establishment. Establishment, where it is registered, the animal house facility, location, that address, institution has to be filled. Okay. So for example, Raipur Institute, Anantapur, complete details. And registration number and date of registration. This is very important. The registration number of the CCSCA registration number, not the institute registration, not the act of the establishment. Okay. So here this number will be presented as the registration certificate provided in the CCSCA, and every 5 years it will be renewed. This institution registration will be renewed every 5 years. So here we have to put the renewal date, not the first time establishment registration is not required. Okay. And name and address of the registration number of the breeders from which you are going to get the animals. So here we have to mention the 1 or 2 or as many as you wish. You can mention the name of the institute where you are going to get the animals for your research experiments, and you have to mention their registration number. Okay, this is important. Their registration number is important, and their registration should be valid. Then only it is acceptable. Okay, if it is expired, they cannot supply, supposed to be supplied. Okay, like that you can mention any number of animals, suppliers. Okay? So registration number is compulsory, and of course the complete address of the animals. The next one is place where the animals are presently kept. So this is also again the establishment where your animal house facility is located. That address has to be provided. Sometimes that address will be within the campus or outside the campus. That facility address has to be given exactly where the location of the animals will be given. Place where the experiments is to be performed. This will be similar. Okay. ఎక్కడైతే animals we kept, that place will be the same as we do the experiment. Here also we have to provide the registration number. The sixth one is important, date and duration of the experiment. Most of the times this will not be filled by the investigator, because this is important, because the investigator does not know when to start these experiments. That is why they kept blank. But it is important, tentative, at least tentative date of start of experiment we have to put, and duration also you have to mention. That is why it has mentioned tentative. Okay. So seventh one is type of research involved. So you should have some idea about what kind of research you are doing, what kind of animals you are doing for what kind of research. Generally we do basic research, basic investigation, okay, basic fundamental research we do, some educational purpose we get some animals, regulatory OECD guidelines ఉంటాయి, toxic effects study చేసడానికి. Some people do contract research. So if someone sponsors the project, they come under the contract research. So most of the time academic institutes, they do the basic research. We have to mention these things. So while submitting, you have to put the date, place, and you have to do the signature. Principal investigator only has to be signed. Okay. So these are the straightforward questions related to the information pertaining to the principal investigator, institute, and supplier of the animals. Okay. Is it clear? Any doubts in this first section? So this is common for all of the investigators who is going to submit, except the name of the investigator. Other things will be very common. Okay, and of course the date of experiments. For example, in your institute, 10 principal investigators are there, 10 proposals are submitted, 10 proposals are approved. That means each one have maybe having 50 animals approved or 100, 50 into 10, 500 animals. Is there any facility in your institute to handling of 500 animals at a day? So in that case, if you don't have the facility to handle 500 animals at a time, you have to change this date. Okay? You have to change the date and then get the animals accordingly. And coming to the section 2, this is very important. It deals with a lot of things. You should be familiar with things. Project title, dissertation title or thesis title. So this is very important. So project title must be complete with the name of the animal species used in the project. No abbreviation should be allowed. So while proposing your title for this, you should mention what kind of animals, rats, mice, even species also here. Within the mice, C57, black sea or bulb sea mice. If it is in the rat, you can star rats, albino rats, all those things has to be mentioned. And also you have to mention the type of compounds you are going to evaluate. Okay? You should give the name of the compound. Broad classification of the name of the compound also has to be mentioned. So this title can be directly used for your PhD work or dissertation work or any other project submission work. Okay? But it should contain what is the animal species you are going to use, and what is the compounds broad classification, what is the name of the animal model you are doing. All those things. I have given an example, therapeutic role of compound X and Y in acute lung injury in mice. So mice species also you can mention, C57 or like. So is it clear? Project title, okay, it should be very clear. It should not be same as your PhD also. PhD title may have different objectives, but one of the objectives should be animal work. So that animal work should be reflected in this. So then only the PhD committee will also accept, even though it is different from the IAC approved proposals. Here the name of the principal investigator should be mentioned. Okay. And as I mentioned, if it is a postdoctoral project, with a specific funding agency, it can be postdoc also can be act as a PA. So So in case of collaborative projects, it is important. Different people may submit the proposals to your institute to get the approval because that institute does not have any animal house facility or IAAC to conduct the experiments. In that case, you have to do a collaborative kind of project. That means that institute, whoever is applying for your institute, has to submit a collaborative letter, and you have to establish a collaboration between that institute and this institute. Then only you can go for submission of the application. In that case also, PI should be from this institution, not from the collaborative institute. PI should be from this institute, and OPI can be mentioned in the next point. Whoever is applying for a collaborative things, that can be mentioned in the serial number. So all these details are very common. Name of the investigator, designation, or other contact information. Here is about experience in the lab animal experimentation. This is very important. Okay, how many years you are having experience handling with the animals? List of individuals authorized to, along with the principal investigator, if it is a B pharmacy project or if it is a M pharmacy project, their names, students' names also has to be incorporated in the third point of the section 2. Okay, main PA along with the other authorized persons. Okay, if they want to publish, at least one of the students' name should be there in the section 2 of the reader. Okay, it is very mandatory to list all the names of the PAs. And also veterinary name also should be included in case of invasive, stressful or painful procedures is involved in the animal experimentation. Because veterinarian is the only person can handle, identify this kind of painful procedures. Okay? Even though the PI is expert in that particular things, but IAS are the veterinarian. That is why we included veterinarian in our IAS. Okay? So important protocols should include veterinarian also. Funding resource, this is also important. Generally people will keep this as a plan. So you have to mention full name of the funding. If you don't have any funding, you can write internal sources of funding. RIFR will provide the funding for conducting this kind of experiment. So these things, you can, if you have the funding agency, you have to attach the proof of that funding agency letter. Okay? So is it clear? Third and fourth. This is also straightforward. But each and every section, the contents has to be filled. It should not kept blank. If it is not applicable, you just write not applicable. That's it. Duration of the experiments. This is also important. Okay, as I mentioned, you have to put the proposed or tentative date of start of experiment and end of the experiment. Okay, so date of the initiation should be after the IAC meeting. Okay, not before the IAC meeting. As I mentioned in the rule 8A, no experiment will be conducted before approval or prior approval from the IAC. So that is why this date should be after the IAC approval only. Don't mention the days. You don't need to mention how many days, all those things. Just month is sufficient and year is sufficient. Describe the details of the study plan to justify the use of animals. So this section is very important, sixth section. So generally you have to attach a separate sheet of paper with all the details. Okay? It should be written in a layman's language. That means in IAAC, scientist will be there, non-scientist person also there, socially aware member also will be there. It should be easily understood by all the members. That is why we have to mention in a layman language. So please provide a brief description of the project, incorporating the background of the project. That is, what area, review of literature, what are the problems you want to do, what are the lacunae that is present, and what are the issues you want to take up. All those things has to be mentioned in the annexion. Okay? Objective of the research, relevance to science and human also. You have to provide the references. So just like that, you should not propose the proposals or projects, you should have the rationale for that. Means you have to refer the literature, there is a problem in the literature, you have to rectify the problems by taking up these experiments. So those things has to be clearly explained in the annexure, in the sixth point. And other one is animals required. So most of the people will get problems in this section. Okay? Animals required for conducting of the experiments. Here we have to mention the species and strain. Many of the people they just mention mice, rats, rabbits, guinea pigs, something like that. But we have to mention the species. C57 black 6 in 1 mice species like that. Bald sheep, okay, DAB mice, all of them. Age and weight. These things has to be decided by the principal investigator after thorough investigation of the literature only. You should not randomly select the animal which will not yield any kind of output. Okay? And number of the animals used in this experiment. So for example, you have 6 groups, okay, or 10 groups. How many mice for each group has to be mentioned. Nowadays, minimum number of animals is allowed is 5. You should not keep less than 5 animals, okay? Or it is always better to go with 6, okay? Even though IAS members ask you to reduce the number below 5, you should not accept as a principal investigator, okay? Because many publications, journals, they do not allow you to go below the 5 number of the animals. So this can be calculated based on the power analysis. We have to use some statistical tools. So what is the exact number of the animals required per group is based on the some statistical analysis or based on the literature. We have to take some of the good impact factor containing journals to decide the number of animals required per each group. So you should see if it is a more variations within the group. For example, the control group, if you see within the animal to other animal lots variations, you should increase the number of animals. If you don't find any much variations, you can stick with 5 or 6 numbers. So variations are important in this case. So like that we have to calculate how many number of animals required to conduct one experiment for 1 year, 2nd year, 3rd year. Depending on the project, some projects may be 3 years, 4 years, 5 years. So you have to break down the animals required for each year. IAEC generally approves the animal experiments for only 1 year. So if you want to extend that protocol, number of animals for the next year, you have to apply it again as a renewal for this project and then take the approval for more number of animals. Okay, generally valid for 1 year of approval. So in this case, you have to define each experimental group, control group, disease group, treatment group, or any positive control group. All those things has to be mentioned. You have to calculate the number. This number has to be clearly mentioned in the certificate and also in the table and also in the annexes whatever you provide. You should not be different from different places. Okay? You have to exercise this very important thing because this number once it is approved it cannot be changed. Okay? When animal suppliers want to supply animals, they generally ask the certificate, approval certificate. Some institutes nowadays they are not asking because of business purpose, but many institutes like Vivo Biotech, NIM, they ask the animal ethical approval certificate. According to that, they will supply the animals. Okay? So like that you make a table. And number of days each animals will be housed in the animal facility. So depending on the duration of the experiment, you have to mention this point. 2 months, 3 months, 4 months. Depending on animal model. Some animal models may be 1 week, some may be 10 months, 2 years, sometimes like that. Okay? And other one is important thing is rationale of or use of animal uses. So 8th point of the section B is rationale for animal uses. Why is the animals use says is necessary for these studies? You have to justify it. Okay? Without justifying the animal uses, IAAC will not be approved. Justification should be why your research questions cannot be answered without animals. So without animals, your problem cannot be solved. That justification you have to give the IAS or you have to write it here. Otherwise, it is not approved. Model relevance. Mention why this specific animal model is important for this study. So there will be several models. Based on the literature study, you have to select the best suitable model for your experiments. These things also important. And no alternative. You have to mention there is no other alternatives than this particular species, this model to conduct or to solve one of the my objectives of the project. And regulatory or translational needs. If applicable, link the study to preclinical requirement. So these regulatory experiments we do. As I mentioned, basic research we do. Regulatory means some companies produce the compounds, new chemical entities, already existing compounds. They have to test it for the toxicity. So for testing of the toxicity, animals also will be used. Okay. So for that purpose, there set of rules are there. OECD guidelines. So according to the OECD guidelines, it is a regulatory requirement. Every compound, before it going into the clinical trials, it has to be tested preclinically in the animal models. Right? So that comes under the regulatory guidelines, OECD guidelines. For that, you don't need to explain much of the things. You just OECD guideline 420. According to that, this many number of animals has to be allowed. That's it. Okay? Translational research for that purpose also you have to mention. So if you give clear justification, what experiment you are doing, alternatives are there or not, how you are going to handle, what species, all those things you provide, that will give you a ratio. Without having things, if you propose, that proposal we call them as irrational. Okay? Irrational proposals will not be accepted. Rational means you have to thorough background knowledge on the experiment what you are doing. You have to link with the existing literature and what you are going to get from the current experiment. That link has to be established. That is called rational of the experiments. Whether similar study has been conducted in vitro models. Animal experiment we call them as a in vivo experiments. If you do outside the animals using cell cultures, tissue cultures, animal cells, so that is called as a in vitro models. So in vitro models are not painful, no distress, nothing, right? So if it is able to conduct the experiments using in vitro models, why you have to conduct the animal models if it is available? So you have to do the justification. Okay? You have to do proper scientific justification. Why you have to use the animals? Okay? If yes, you have to provide the literature. Why are the particular species is selected? For example, you take mice, rats, guinea pigs. You should give the proper justification. Mice or rat or guinea pigs are very established model to study this kind of disease based on the literature survey. Okay? Some animals are genetically available to test for some kind of diseases. So we have to select based on the literature and what kind of output, what kind of results you are going to get. So that species justification has to be given. Why is the estimated number of animals? Already mentioned. Okay. So this number is can be get from the different statistical tool. Okay. G power, Crawford prism, based on the literature. You have to mention the number of animals, estimated numbers, based on the statistics or based on the already existing literature. Okay? You have to add the references also to justify this many number of animals, this many groups have been included in the protocols. So that it is easy for IASC to scrutinize and give the approval for the project. Are similar experiments conducted in the past? If it is already conducted some kind of experiments on the same compound or same animal models in your institute, why you are doing this kind of experiment again? It is already conducted this kind of experiment. Straight away it will be rejected by the IASC. You have to give the justification. If the project is a continuation of the laboratory, so animal model is same, but you are testing with different compounds, different formulations, different chemical entities. nano formulation, whatever it is. You have to give that justification. That work has not been conducted. That is why, even though this model is established in this laboratory, we are using a different colors. Okay, so those things we have to mention. Experiments. Does any other organization conducting similar kind of experiments? If they are conducting, why you are conducting here? That justification also you have to give. So many of the works, for example, animal models for neurological diseases, lung diseases, cancers, cardiovascular diseases, are doing in different laboratories. In Kadapa, in YBU, in this Raipur, or CCMB, or IAC. Many laboratories is conducting same animal models, but their objectives are different. Some is studying basic pathogenesis, some trying for new chemical entities, pharmacology studies, pharmacodynamics studies. Different studies they are doing. Okay? So for that purpose, you can write several groups in India or abroad working on the similar models, but not with the similar compounds or pathways to understand. So this kind of justification you have to give. Generally, we are not going to develop a new model. Most of the time we are using the same animal models for our research also. But only thing is we are working with different compounds or different pathways we are studying. So that justification has to be given. So part 8 is very important. That is rationale of usage of animals. Animals ను మనం ఈ రీసెర్చ్ కోసం ఎందుకు వాడుతున్నాం అనేది క్లియర్ జస్టిఫికేషన్ ఇచ్చే పాయింట్ ఏంటంటే 8. Why animals are research important? Why these species are? Why the number of animals you propose? Is there any person doing the similar kind in your lab or other labs? All these things has to be justified properly. If you want to do this kind of justification, you have to have a thorough knowledge on the subject what you are proposing. In the last minute, if you come and submit applications, you will miss all the information. You are just copying pasting of the some other experimental protocols and submitting. While scrutinizing, we will see same proposal, same number, same animals. The title will be different, animal proposal will be different, methodology will be different. All these issues will be raised when you don't have thorough knowledge or proper exercise on these particular things. Most of the investigators or students are least bothered about Form B. We have a lot of experience by scrutinizing this kind of Form Bs because I was a member for more than 20 institutions as a nominee, scientist, social event. Most of the time we see missing information or wrong information because copy paste. Because principal investigator or the guide will ask the students in the last minute, IAC meeting conducting, you please submit your applications. So they will submit within 2 days based on the previous protocols and other things. They do a lot of common mistakes. So you have to decide what project you want to do earlier stage of your research. Okay, then only you will come up with good proposals. Does any organization take it right? So next one is 9th point of the Form B section. Describe the procedures in detail. So this is also important. This part, okay, mainly deal with minimization. 3 are principles, right? Minimization of the pain, distress, and also suffering of the animals. So if you follow this protocol sufficiently, you can do reduction in the pain, suffering, and the distress of the animals. All the animals used in the study undergo some minimally invasive procedures such as oral, intraperitoneal, administration, exposing animals to different chemicals, different inductions, right? So invasive means you are injecting something inside the animals. Non-invasive means like behavioral study. We get the animals, keep there and observe the behavior of the animals. That is called non-invasive. Most of the non-invasive experiments, animals will generally not suffer. Okay? When you handle the animal, when you inject something, they feel stress. They suffer. Okay? So for that, most of the experiments are invasive procedures. At least we give drug, right? Drug orally. If you want to give drug, you have to restrain the animals. You have to restrain the animals, hold properly. Animals ఎలా behave చేస్తాయంటే, if you are keeping animals for more than 1 month, the same person is going handling, it will not feel any kind of stress. If any new person go and handle, immediately they get the stress. Your experimental results will be changed. So that is why acclimatization for 1 week, 10 days is very important. Okay? When you get the animals, keep it in animal house, wait for 1 week, 10 days and see the behavior of the animal. If everything is good, then you start the experiment. Experimenting person who is handling the experimenting person should be familiar with the animals, like how you are familiar with your kids and neighbors. You should be familiar with the animals. Then only you can get expected results. Otherwise, you get nonsense results which cannot be expected. It cannot be utilized for any of the papers also. So that is why it is very important. Details of the injections or substances you are going to give. Doses. Test compounds like drugs, amoxicillin, hormones, or whatever substances you are giving. Route of administration, orally, peritoneally, intratracheally, okay? Whatever route, that route has to be mentioned, okay? And the doses also clearly has to be mentioned. This dose and route has to be exercised based on the preliminary work or based on the existing literature. Most of the chemical compounds are pharmaceutical compounds which has not been established toxicity evaluation. So you have to done. Okay? Toxicity has to be performed first. Then only you have to decide based on the IC50, LC50 value, you have to decide the doses. And you have to give proper references also where you have get the dose combination, route combination. And this is also important, volumes. Volumes generally, if you give orally for mice, 200 microliters will be given. If you are doing intratracheally, opening of the trachea and injecting the drug into the lungs directly, you have to reduce the volumes like 10 microliters, 50 microliters maximum. Or if you are given intranasally, you have to reduce the amount, 10 microliters, 20 microliters. So while giving the drug, for example, if you are giving the drug intranasally, you have to partially anesthetize the mice. Otherwise, it will be squeezed out. The drug will not be entered into the mice. Or if you are doing intratracheally, you have to anesthetize the mice. Orally, it is not required. Okay? So depending on the route of administration, volume of the drug you are going to give, you have to decide whether you have to anesthetize the mice to relieve the pain and proper injection. You have to decide. All those things are important. So to avoid the pain, distress and other things. So every experiment, most of the experiments, it is invasive and then it requires collection of blood tissues from the samples, right? So most of the time we take the blood. So blood we analyze for different parameters. We can analyze the count, cell counts, cytokines, number of proteins, antibodies. So many things can be diagnosed using the blood, right? So that is why it is very essential things to get the blood from the animals after experimentation or during the experimentation. So this you have to mention, depending on the species, how much volume maximum can be connected without euthanizing the animals. Okay? After euthanizing, what is the animals? Where you are collecting sites. Okay? So you have to indicate the method of collection. For example, I have given the table here, the reference purpose. Mice, by tailing you can get maximum 100 microliters, small volume. Okay? If you want to get more than 100 microliters, you can do retroorbital sinus. Okay? Without killing the mice. First two. If you want to get more, you have to euthanize the mice. And then you do the cardiac puncture. You can get maximum 1 ml using the mice. Like that, rats, there is a specific routes and sites, volumes also. Rabbits. All these are the small animals. Small laboratory animals. There are two classifications in the laboratory animals. One is small animals and the other one is large animals. Small animals, experimentation can be approved in the IAC of the institute. If you want to do large animal experiments using dogs, monkeys, non-human primates, okay, you have to take the approval in the institute from the IAC and then the same has to be submitted to the SEA for further approval. Okay? For that, larger animal facility also has to be registered. So this institute, RIPER, does not have the permission to conduct any experiments on large animals like dogs or beyond that. Okay? For that, we have to follow the same protocol, but it has to be finally approved by the CCS. Okay, every 3 months they conduct the meetings there. You have to submit that, get the approval, then only you have to do the experiment on large animals. The facility also has to be registered. And facility should be registered for two things. If you want to do basic research, just registration is sufficient. If you want to breed animals, you have to have the different registration number. So this institute does not have any registration number to breed the animals. Only get the animals, do the experiments, finish it. Okay? So that kind of rules you have to be familiar with the things before you start any kind of experiments. Nature of the compound. So this is important. Recently included in the Form B. Previously it was not there. So you have to mention in the title itself what is the nature of the compound you are going to test. So nature means whether it is a chemical compound, whether it is a biological component, you have to mention that comes under the nature. Natural products, synthetic products, biologics, vaccines, gene therapy, vectors, all those things are the one class of compounds. Broad classification. Classifications means biologically, immunomodulator, antioxidants, anti-inflammatory, antioxidants of so and so compound will be tested in this model using this mice rats. So that entity you can include in the title itself, or you have to mention here natural compounds. Okay, classification anti-inflammatory. It is a new chemical entity. New chemical entity means which has not been approved by the FDA or other agencies. If you are testing, you have to provide those agencies. Okay? So this is only for internal purpose. You should not be maintain confidentiality also, because you should not be carried forward to the other institute. IAC members working in different institutes should not carry the information, because sometimes it is linked with some kind of the patent issues also. So those things has to be kept in mind while mentioning the new chemical entities all over. The 10th part is, does the protocol prohibit use of anesthetics or analgesics? So these anesthetics or analgesics are very important because it will alleviate the pain of the animals. Okay? You are doing animal experiments, you are doing something, if that causes the pain, you can give anesthesia while doing, or after that you can give the analgesics. Okay? When your protocol does not affect these things, okay, you should give any one of these to reduce the pain, suffering or distress. If it is inhibiting your outcome of the project, you should not give these things. Okay? So that you have to mention clearly here. This study does not prohibit the use of anesthetics or analgesics. Hence, you can give the, for example, you have created wound, that wound gives some pain to the animals. You can give analgesic during the process of evaluation. That way, if that is not affecting the pathway that you are studying. Okay, so these kind of things also you have to mention clearly in your protocol. So this is again is based on the literature. Okay, literature study cheyali. Otherwise, you have to have the prior knowledge on that particular model. This is also important. Survival surgery. Will survival surgery be done? Means when you are doing experiments with animals, you do some surgery. After surgery also you are maintaining animals in the laboratory. That procedure will be called survival surgery. If you do survival surgery, you have to maintain the animals in the laboratory for some time, 1 week, 10 days. For example, in our laboratory, we give the anesthetics to animals. We induce the lung injury by intratracheal injection. So while doing intratracheal injection, we anesthetize the animal, we dissect the animal, and we open the trachea, inject the animals, and we make the sutures, and then that animal will be survived. Okay? Like that we do cecal ligation function, sepsis, microbial sepsis model, cecal ligation function. For that also we open the abdomen, take out the cecum, puncture the cecum, and tie with knot and then put it back, the cecum inside and we make the sutures and the animal will be survived. That kind of models we call as survival surgeries. So when you do the survival surgery, you have to use proper anesthetics to avoid the pain during the surgeries. And the person has to be qualified, experienced. Without having any experience, if you perform, the animals may die. Okay? Anesthetics also, if you do overdose, less dose, they will feel pain or they may die. So depending on the experience of the person, we have to select the thing and do the things. Post-operative care. After doing the surgery, you have to do post-operative care. You have to give antibiotic or analgesics. You have to maintain proper conditions in animal house, like how we do for human beings after surgery. Similarly, we have to do the animals also, otherwise you don't get results. Just for sake of projects, for sake of thesis, if you do experiments, you don't get any results. Generally what they do, even though they don't get results, they put something else and then they get the publications or whatever thesis. That is not good. Okay? So that is why Form B is mainly made for, you have to justify scientifically, first thing. And second thing is animal welfare. Those two important things you have to keep in mind when you do experiments using animals. Scientific justification and animal welfare. Morning చెప్పిండు సార్ మీకు animal welfare. Animal welfare issues చాలా ఉంటాయి. ఓకే, మనం ఇంట్లో పెంచుకునే డాగ్స్ సఫరింగ్ లాట్, మనం తెలియకుండానే సఫర్స్ లాట్, దాని నార్మల్ బిహేవియర్ అంతా లాస్ ఉంటుంది. అలాగే అనిమల్స్ కూడా నార్మల్ బిహేవియర్ లాస్ ఉంటుంది. అందుకని మనం కేజుల్లో సోషల్ ఎన్రిచ్మెంట్ డివైజెస్ పెట్టాలి. ఆడుకునేదానికి, ఎగిరేదానికి, పైకి పోయేదానికి, ఆగేదానికి అన్నిటికీ సోషల్ ఎన్రిచ్మెంట్ డివైజెస్ అంటారు. వాటి మన కేజులోనే పెట్టేస్తాం చిన్న చిన్న ఐటమ్స్. సో దోస్ విల్ హెల్ప్ టు రిలీవ్ ద స్ట్రెస్ ఫ్రమ్ ద అనిమల్స్ so that you get the good results as an outcome. So these are the normal generally used anesthetics. Isoflurane is a gas, ketamine gelatin combinations, sodium thiopental, and the pharmacy all these things. Okay, so you have to select based on the things. Some anesthetics have some effect on the muscle relaxation and other things. If you are doing some kind of experiment using on muscles, you should avoid that have the effect on that things based on the literature. Post experimentation procedures is also important. Scope for reuse. Scope for reuse means if the animal experimentation does not require euthanasia, killing of animals. Okay, experiment అయిన తర్వాత అనిమల్స్ ని చంపకుండా మనం ఏదైనా ఉంచగలిగితే వాటిని యూజ్ చేసుకునే కెపాసిటీ ఏమన్నా ఉందా? Most of the time small animals, laboratory animals like mice, rat, guinea pigs, hamsters will not be reused. Okay, will not be reused. For education purpose, handling purpose, they may be used, but experimental purpose generally it is not recommended. Small animals. Rehabilitation, rendering of the aftercare of the experimentation. For example, mice, rats, we don't rehabilitate. In small animal categories, only rabbits will be rehabilitated. Okay, rabbits మాత్రమే రీహాబిలిటేట్ చేయాలి. ఆ రీహాబిలిటేట్ చేసేదానికి సమ్ ఏజెన్సీస్ ఉంటాయి, లేదంటే మన దగ్గరే ఫెసిలిటీస్ ఉంటాయి. న్యాచురల్ డెత్ అయ్యే వరకు ఆఫర్ చేసే ఫెసిలిటీస్ అన్నింటినీ కూడా ఆ పర్టికులర్ అనిమల్స్కి ఆఫ్టర్ ఎక్స్పెరిమెంటేషన్ రీహాబిలిటేషన్ అంటారు అన్నమాట. So name and address. For example, if you are not able to rehabilitate animals after experimentation, particularly rabbits, for rabbits also comes under the small laboratory animal. Okay, small laboratory animal is rabbit, it looks little big. Okay, వాటిని కంపల్సరీగా రీహాబిలిటేషన్ చేయాలి. If you are using rabbits for some experiments for monoclonal antibodies or polyclonal antibody generations, after experimentation we only collect the blood, we will not euthanize the animals. After that you have to rehabilitate. That is specifically you have to mention. Describe the method of euthanasia. Humane methods under human methods of sacrificing the animals. Most of the time we write sacrificing of the animals. It is not a good word to use it. Animals are not, we are sacrificing them. They are not sacrificed for us, right? Sacrifice means something we are giving up. So generally, euthanization, God is good for that. So generally we give 3 times or more than that of overdose of some anesthetic, or if you have the CO2 cylinders, you put animals in a small bag, supply the CO2, and then within a second the animals will be killed. Okay, so that is acceptable methods. Some methods will influence the outcome of the experiments. We have to select a suitable method for euthanasia. Okay. Sometimes people will do cervical dislocation. Cervical dislocation is very commonly used. So during that, lot of bleeding, lot of pain will be seen. So that should be generally avoided. Okay. For that purpose, you just give anesthesia, small doses, and then after anesthetic unconsciousness, you can do the cervical dislocation. So all these things are important for euthanization purpose. The last one is method of carcass disposal. After experiment, anything left over animal carcass, how you are disposing? There is a bio-waste medical management system are there. Pollution control, central state pollution control boards are there. You should not dispose whatever you want like that. Okay? So there should be a proper disposal. So for that, after animal experimentation, whatever the carcass is present, that has to be stored at refrigerator for some time before it is collected by the biomedical waste collection agencies. Okay? Or if we have an incinerator means the device that can be used to burn the carcasses. So this can be purchased, so it will avoid lot of issues when you do have this kind of incinerator. Otherwise, you have to submit the waste to the biomedical agency suppliers. Also, on the contract value. These things have to be mentioned clearly in the proposal. Okay, most of the times this section will be ignored because you don't have idea how to discard. And other one is, deep burial ground lo manamu tobesi pettastunnam. If you don't have the incinerator, if you don't have the biowaste, the last option is the burial ground deposition. Okay, last option. That sometimes it is not acceptable. It has to be compliant with the pollution control. Pollution control to acceptable like the deep burial. So this has to be discussed with your institution before you write whatever the comment here. Animal transportation. This is also important. So if you are purchasing animals from somewhere like Hyderabad or Bangalore, this has to be transported in air-conditioned vehicles. Sometimes they will fly also. Okay, in a specific, containers which has the proper caging, bedding, feed, water during the transportation. Okay, laboratory animals ki there is a specific cardboards are available with proper ventilation, bedding. So those kind of cages has to be used while transporting. All the time it has to be put into the air conditions. If the temperature goes beyond 25, 30, 40, some animals may also die. Okay? And even if they don't die, they suffer with lot of diseases and you don't get the expected results when you do the experiments. So these things you have to keep it in mind while transporting the animals. Hazardous agents. So when you do experiments, any protocol, any substance has any kind of these things. For example, if you use recombinant DNA molecule or recombinant proteins you are injecting, or you are injecting some kind of pathogens like virus, bacteria, microorganisms, it has to be approved by the institutional biosafety. There is another committee besides the institutional animal ethics committee. There is a committee called the institutional biosafety committee. This committee has to be approved and they will give the certificate. That certificate has to be attached with the IAC forms. For example, when you are doing work with radioisotopes, okay, radio labeled ATP, radio labeled proteins, radio labeled ID molecules, you have to have the permission from the Atomic Energy Regulatory Board, board, all those things, okay. That certificate has to be adopted. IBS as mentioned. If you want to use recombinant DNA proteins, Review Committee of Genetic Manipulation certificate has to be there. Okay? Chemicals, drugs, pollution control, all these things. Okay? If it is involved, you have to attach a copy of the approval, prior approval. Okay? Without approval of these things, this kind of experiments cannot be used. Okay? So this is all. If it is nothing is involved, you just mention not applicable. Okay? Just mention not applicable. Your drug or compound does not come under any kind of these regulations. That is why you can get the not applicable. And this is the last page of the certificate, last page of the Form B. This is the investigator's declaration. Means after filling of the Form B, the investigator has to be go through these things and has to be signed. What it says, the research proposal submitted is not unnecessary a duplicate. That means he has certified all these points. It's not a duplicate. I certify that I'm qualified doing these experiments, and also it has to be approved by the IAC before conducting the experiments, and relevant experiments is selected for these things as per the regulatory bodies, and also certify appropriate certification from the review board. For example, as I mentioned, radionuclei or microorganisms we are using, all those things as mentioned in the point 14. I shall maintain the records of the Form B, Form D. I mentioned the different forms in the previous slides. So once you do the experiment, you have to maintain the Form D. All those things you have to confirm, you have to declare that I am following these rules. He has to certify. If he certify that, he is responsible for any deviations he do. Okay? And if any deviations, he is punishable, offense. Okay? So that is why this certificate has to be carefully read and signed. And this is the final certificate will be issued by the IASC after approval of the animal experimentation. So here they will mention the protocol number. This number is important. This number is a serial number of your institution where year-wise, PA-wise numbers will be given, and this number is important for publications also. If you want to publication, the publishers ask for the IASC certificate. So for that purpose, you can give this number. And PA name will be there, title will be there, and the name of the institute, meeting head, and how many number of animals has been approved, and how many months duration it has been approved. So this is the brief information to be provided in the certificate, the red colored. And it has to be certified by the 3 persons. One is the chairman of IASC, other one is member secretary. These two come from the same institute, and main nominee is nominated by the CCSCA. He will always be present in the every meeting. He has to be certified. Then only it is the ethically approved and you can conduct. After getting this certificate only, you have to perform the experiment as per the rule 8A of breeding and experiments prevention. Okay. So this number, all these things has to be mentioned in the minutes of the things. So I have 2-3 slides. Okay. These are important things. You just please be carefully listen. Tips for the scrutiny and approval. Submit Form B along with request letters to the IASC well in advance. Generally, last minute submissions have a lot of issues. Okay, at least 15 days advance, you have to prepare and submit to the IASC members so that they will be scrutinized applications. If any queries are there, they will be sending back, okay, to the PIs and for rectification. So generally, PIs should not be available for presentation. Only whenever there is a queries, they will be called. Otherwise, there is no formal presentation required. If you submit well in advance, at least in 15 days, so that they have the chance to read it. So align your proposal with 3 R principles. I already explained what are those. So this is very important. Many of you are not aware of this. Arrive checklist. So arrive checklist has been latest entity in the ethics. I will come to that. Use clear, complete language. No points in copy should be unanswered. If it is not related, you just have to mention the not applicable. So this arrive guidelines, animal research, reporting of in vivo experiments. So these are the guidelines started in 2010. Okay, international guidelines. So these guidelines ensures scientific rigor and animal welfare. So the regulatory body in India is CCSCA. Outside the India, there are several regulatory bodies that take care of scientific validity and animal welfare issues. One of that is Arrive guidelines. So without this, many of the international journals are not accepting papers. So this has two sections. Okay, this is the website for the Arrive guidelines. It has Arrive essentials. It has 10 points. So that 10 points, as I mentioned in the IAAC Form B, some of them are covered. Those are considered as essential. It is compulsory. Some of them are recommended, like our animal facility, bedding facilities, air conditioning facility, water, all those things. So this has to be completely checklisted and submitted along with these papers to the journals. Then only people are accepted. So that is why while doing Form B, you also have to check the Arrive guidelines and you have to be thorough with guidelines or checklist while submitting the Form B without any rejections. So common errors should be avoided. Incomplete group details, statistical justification, if it is not there, it will be rejected. Lack of humane endpoints like proper anesthesia for euthanization has to be utilized. Copy paste of other PA student information should be avoided. Poor handwriting of physical form should be avoided. Okay, all these things are the common errors people will do. So if you avoid these things, it will be easily approved. So final conclusion is Form B is not just a format. It is not a bureaucratic setup. It is ensures the ethical, scientifically valid animal research. If you want to do ethical research, scientifically valid research, animal welfare also should be considered. All those things will be covered in the Form B. That is why be transparent, precise, and humane when you work with animals for research purpose. I hope you understand the different components of the Form B. Section 1, general information. Section 2 is information related to the experiments, PA experience, number of groups, how you are euthanizing, pain, distress, how you are avoiding. All those information is given with proper suitable examples. Okay? So now I will open this all for any kind of questions related to this Form B field. So I thank the organizers for giving me this great opportunity to cover some of my experiences in filling up the Form B as a IAAC nominee for the last 15 years. So we see a lot of problems in the IAAC forms. So most of the issues, whatever we generally see in the Form B, has been explained properly. And I will share this PPT along with the Form B so that you can go through that. And it is very straightforward. If you are very clear in your project, this can be easily hit. So once again, I thank all the participants for nice listening my talk, and I wish all the best. Any questions? Questions లేవా? ఎప్పుడన్నా సబ్మిట్ చేశారా ఐఏఎస్ ఫార్మ్స్? లేదా చేస్తారు ఫ్యూచర్లో? You want to become a good scientist, you want to use animals for research purpose, you should be familiar with these things. Animal welfare, scientific validity, most important things you have to be kept in mind. No questions. I will stop here and I will hand over the mic.
Yes,